This is Professor Doherty, and today we're going to be looking at Lab 23, which involves looking at cortisol levels within saliva. Now, first let's talk about what cortisol is. Cortisol is a steroid hormone. It's produced in higher levels during times of stress, so it's considered to be a stress hormone. It's actually made in the adrenal cortex, and it has a number of different functions. The primary ones that I think of when I think of cortisol is it is a glucose regulator, as well as it can suppress the immune system. To test for cortisol levels in saliva, today we will be using something called an ELISA test. This stands for Enzyme Linked Immunosorbent Assay. Before we get started with our lab, first let's review how an ELISA test actually works. The first thing that we need to do is identify our target molecule. In this case, our target molecule is human cortisol. The next thing we need to do is develop an antibody to that target. The way that we would typically do this is we would take human cortisol and inject it into another animal, for instance, a rabbit. That rabbit will then develop antibodies to the human cortisol, and we can then harvest those antibodies from the rabbit's blood. Next, we would need to develop a form of cortisol that has an enzyme linked to it. Now, this is going to be a competing form of cortisol that will compete against our salivary cortisol. Finally, we will add a substrate to our solution. This substrate is designed to react with the enzyme, and when they come together, the enzyme will cause the substrate to give off a color. And depending on the intensity of the color, we can determine the amount of cortisol that was originally in the saliva. Now let's take a look at this procedure in a drawn format. So here we have an ELISA well, and the well is coated with the antibodies to cortisol. We will then place a measured amount of saliva and a measured amount of enzyme attached cortisol into the well. These two forms of cortisol will then compete for sites on the antibodies, and after a designated amount of time, we will then wash away any non-antibody bound cortisol. We will then add a substrate into the well. This substrate is going to react with the enzyme to create a color, in this case the color yellow. At the end of the experiment, the more intense the yellow color, the more enzyme-linked cortisol we have in the wells. Therefore, the more intense the color, the less salivary cortisol was present. So, more color means less salivary cortisol, less color means more salivary cortisol was present. Now that you have an understanding of how ELISA tests work, we can now discuss the procedure for today's lab. I will be starting this section on page 3 of your lab handout. We will start the lab by making our standards. This consists of 6 wells that will each receive a known concentration of cortisol. The wells will be numbered from 0 to 5, with well 0 receiving no cortisol and well 5 receiving a 30 nanogram per milliliter concentration of cortisol. The well numbers and standard concentrations are listed in the chart on page 5 of your handout. We will also be making one experimental sample that will receive saliva that has an unknown amount of cortisol in it. Just so you are aware, here are some images of ELISA wells. They typically come in 96 well sets, and the inside of the wells come pre-coated with the cortisol antibodies. Now to begin, as you can see on page 3 of your lab handout, we first add 50 microliters of each of our standards to each of their respective wells. Here I've also listed the concentration of cortisol in nanograms per milliliter for your convenience. Next, we add 50 microliters of the competing cortisol with attached enzyme to each well. Remember that this cortisol that has the enzyme is going to compete with our standard cortisol that we've just added. We then incubate for 60 minutes at room temperature. After an hour, we then pour out the fluid from each well and wash the wells 3 times with 300 microliters of wash solution. Remember that since we have antibodies adhered to the sides of the wells, and we now also have cortisol that is stuck to the antibodies, washing the wells does not remove the antibodies, nor does it remove the cortisol that is adhered to those antibodies. Next, we add 200 microliters of substrate to each well. We then incubate in a dark drawer for 30 minutes. Remember that our substrate will react with any enzyme that is bound to our antibodies in our wells to produce a yellow color. Finally, as our last step, we add 50 microliters of stop solution to each well. This stop solution will stop the enzyme and substrate reaction, so we will get no further color changes in the wells. In the end, our standard wells will range in color from dark yellow at well 0, which has no cortisol, to a clear color at well 5, which received a high amount of cortisol. We will then do the same procedure for our experimental well, but instead of receiving a standard, this well will receive 50 microliters of saliva that has an unknown concentration of cortisol in it. The procedure for the experimental sample is on page 4 of your lab handout. This procedure is exactly the same as the procedure for making the standards, with the exception that we're using saliva instead of a known cortisol concentration. Then for our final step, we will visually compare the color of the experimental well to the standard wells to see which it most closely matches to. In this case, the test well most closely resembles well number 3 of our standard set, indicating that the saliva sample contains approximately 1.7 nanograms of cortisol per milliliter of solution. Now that you have an understanding of how this experiment works, we can now move into the actual experiment itself. First, I should apologize for the angle of the camera on this video. I thought the camera was shooting further down than it was, but you can still see what I'm doing in this video. Just so you know, I will be following the steps on page 3 of your handout if you'd like to follow along. I will also be performing the procedure on page 4. They're nearly identical. It's just that the procedure on page 4 is for the experimental well, whereas the procedure on page 3 is for the standards. So I'll be doing them side by side. To start with, I'll now be setting up the 6 ELISA wells for the standards. I start by pushing them into some yellow clay. This is just to keep the wells from tipping over. I then label the wells from 0 to 5. And then next, I add 50 microliters of each standard into its respective wells. The standard comes in these small vials that are labeled with concentration of each sample. I've sped up the video in certain places of me adding standards or other fluids for your convenience. Next, I'm now adding 50 microliters of cortisol with the attached enzyme to each well. I then tap the wells to mix them thoroughly. I then let them sit for an hour, and while I'm waiting, I then prepare the experimental saliva sample. Here I've already placed saliva in a microcentrifuge tube and spun the sample to remove the air bubbles. I then take 50 microliters of saliva and place it into an ELISA well. I then add 50 microliters of cortisol with enzyme to the experimental saliva well and let it incubate for 60 minutes. So all of the samples will be incubating for an hour. After an hour is over, I then dump the fluid contents of the standard well onto some tissue paper and wash each well 3 times with wash solution. We then add 200 microliters of substrate to all the standard wells as well as to the experimental well. Remember that our substrate is what's going to react with our enzyme to create a color. Then we incubate both the standards and the saliva well in a dark drawer for 30 minutes. This is allowing the substrate and the enzyme to react with each other to create a yellow color. After 30 minutes, we then add 50 microliters of stop solution to each well in order to stop the reaction between the enzymes and the substrate. And this will help to maintain our color. Finally, we compare the color of the well that received saliva to those of the standard. Here we have our newly created standards labeled 0 to 5 with the amount of cortisol in each standard shown in the inset table. Also, we have our experimental well that had saliva placed into it, which contained an unknown amount of cortisol. When we compare the intensity of the yellow color of the saliva well with that of the standard, you can see that it most closely matches standard well number 3. And when we check well number 3 against our cortisol concentration chart, you can see that this indicates that the saliva from our subject contained approximately 1.7 nanograms of cortisol per milliliter. Normal cortisol levels have a range. In the morning, cortisol ranges from about 4 to 9 nanograms per milliliter, which would match well number 4. As cortisol levels drop into the afternoon, they drop to levels of about 1.2 to 3 nanograms in the afternoon, which equals well number 3. Then in the evening, cortisol levels drop to a lower level of about 0.5 to 2 nanograms in the evening, which would match well number 2. Our saliva sample was drawn from our subject around noon, so we would expect our experimental well to match the coloration of the afternoon cortisol sample, which is in well number 3. And in fact, that is what we see. So our experimental results match what we would expect to find based on the clinical observations of cortisol levels. This concludes Lab 23. You should now be able to proceed with answering the questions in the remaining part of your lab. Thank you.